12th Bio-botany Chapter 4 Principles and Process of Biotechnology. In this part, under the heading Molecular Techniques, we are going to discuss about isolation of genetic material and gel electrophoresis. Electrophoresis, electro means electric field, phoresis migration. Electrophoresis is a separating technique used to separate different biomolecules with positive and negative charges. Positive and negative charge biomolecules can be separated from the mixture of molecules with the help of electrophoresis. While applying electricity, the molecules migrate according to the type of charges they have. According to the type of charges they have, the molecules migrate. If the molecules have positive charge, they will migrate towards negative electrode. If the molecules have negative charge, they migrate towards positive electrode. Positive cations migrate towards negative cathode. Negative anions migrate towards positive anode. DNA is negatively charged molecule, so it will move towards anode. Electrophoresis is used for the separation of different biomolecules, but for the purpose of separation of DNA, agarose gel electrophoresis is used. It is used mainly for the purification of specific DNA fragments. From a group of DNA fragments, a particular DNA fragment can be isolated by gel electrophoresis. Agarose is a convenient for separating DNA fragments ranging in size from few hundred to about 20,000 base pairs. Agarose gel is used for the purpose of separating DNA fragments of few hundred base pairs to 20,000 base pairs. For smaller DNA fragments, another gel is used. Polyacrylamide is preferred for the purification of smaller DNA fragments. It is a polymer. Acrylamide is a monomer. After polymerization, polyacrylamide gel is formed. It is used for the purification of smaller DNA fragments. Agarose is a polysaccharide generally extracted from certain red seaweed. Agarose is used as a medium to separate molecules because of its porous and spongy matrix. Agarose is a porous jelly. It has pores. Through the pores, DNA molecules can move, and it has spongy matrix. How to prepare agarose gel? Agarose powder is mixed with buffer. Buffer is something that maintains pH. The agarose powder is mixed with buffer and boiled. When the temperature is coming down, the agarose solution is poured into a mold. Mold is a small box like something. The size of the mold depends on the size of the agarose gel we need for the experiment. After pouring the cool agarose solution, a comb is inserted. On further cooling, the agarose becomes solidified. It becomes semi-solid. Its rigidity increases. When we remove the comb, wells are created. The wells are meant for the purpose of injecting DNA samples. This is a finished gel. The finished gel is kept inside the gel chamber in the electrophoresis apparatus. It has negative electrode, positive electrode. The finished gel is kept here. It is going to be kept. After keeping the gel inside the chamber, buffer solution is poured. Now buffer solution is poured here. The gel must be immersed inside the buffer solution. What's the use of this solution? Buffer has dissolved solutes. Its conductivity creates uniform distribution of ions from one electrode to other. This buffer maintains uniform distribution of ions from one electrode to another electrode. We cannot use distilled water like substance because the solution must conduct electricity. Then only the molecules will move towards a particular electrode. Then the DNA samples are injected into the wells with micropipette. This is a micropipette. Now the second well is filled with DNA sample. So all the wells are filled with DNA samples, and then it will be closed. Wires will be connected. When it is switched on, the DNA fragments start moving towards the positive electrode. The moment of DNA bands are seen as bands. The phosphate backbone of the DNA molecule is negatively charged. Therefore, when placed in an electric field, DNA fragments will migrate to the positively charged anode. Usually, a marker DNA sample is injected into one of the wells. What is marker DNA? It is a DNA sample. We know the length of the fragments within the sample. In electrophoresis, smaller fragments move faster, and larger fragments will cover small distance. This is the longest fragment containing 1200 base pairs. This is a marker DNA, shortest marker DNA with 100 base pair. So these are sample DNA molecules. If we have to calculate the approximate length of this DNA fragment samples, we have to compare the samples with the marker. So this is second marker DNA fragment. Its length is 1000 base pairs. So the sample A, this DNA band has 1000 base pairs, and this one matches 800 base pairs, and this one matches 600 base pair marker DNA. This one matches 300 base pairs marker DNA. This one 200 base pairs. This one matches the smallest marker DNA with 100 base pairs. The electrophoresis is frequently performed with marker DNA fragments of known size, which allow accurate size determination of an unknown DNA molecule by interpolation. The bands of DNA in the gel are stained with the dye ethidium bromide. Ethidium bromide will be added with a buffer solution, or will be added after the bands are formed to stain DNA. The ethidium bromide is absorbed by the DNA, and the bands are illuminated by UV light. DNA can be illuminated in UV light, and the UV light illumination makes the bands visible. This will give orange fluorescence. Orange fluorescence represent the DNA molecules. The light eliminates ethidium bromide. Ethidium bromide shows fluorescence. Ethidium bromide is absorbed by DNA, which can be photographed. The bands can be photographed. Agarose gel electrophoresis is most commonly used in the separation of DNA molecules. The separated DNA bands are often used for further procedure. So many DNA bands are seen. The needed DNA fragments. A DNA fragment may be cut off the gel as a slice. The needed DNA fragment may be cut out of the gel as a slice and then dissolved and purified. This will be used for further procedures. In the next part, we shall discuss about blotting techniques.